Hemoglobin proteins in our red blood cells have the important job of transporting oxygen from our lungs to the rest of our body, so that it can be used in cellular respiration. Once oxygen molecules have been picked up by hemoglobins, there needs to be some sort of mechanism to undo this bond, because our tissues won't be able to use the oxygen if hemoglobin keeps holding on to it forever. Let's talk about the factors that influence oxygen affinity for hemoglobin proteins. This is the Oxygen-Hemoglobin Dissociation curve. The x-axis is the partial pressure of oxygen, or how much pressure is exerted by oxygen molecules in a mixture of gas. It can generally tell you how much of that particular gas makes up the entire gas mixture. The y-axis is the percentage of hemoglobin saturation, which tells you the percentage of heme groups that have an oxygen molecule bound to it. At this point on the graph, hemoglobin saturation is around 50%, meaning that around half of those binding sites are occupied by oxygen. And looking on the x-axis, we can see how much partial pressure of oxygen is necessary to get to 50% saturation. This is sometimes labeled as P50. Partial pressure of oxygen is an example of a factor that makes it more likely for bonds to form between oxygen molecules and hemoglobin molecules. As hemoglobin molecules travel to the lungs, where there are a lot of oxygen molecules around, interactions between hemoglobin and oxygen are more likely. In other words, they are more likely to bump into each other, which increases the chances that bonds will form. As the hemoglobin molecules leave the lungs and travel to areas of the body, such as in the tissues of the legs, with fewer molecules of oxygen or lower partial pressure of oxygen molecules will be more likely to leave the hemoglobin. You can see this being represented with the Oxygen-Hemoglobin Dissociation curve, where a lower partial pressure of oxygen is related to a lower percentage of hemoglobin being saturated. The other factors work by decreasing or increasing the hemoglobin's oxygen affinity. That means that these conditions cause hemoglobin saturation to occur at either a lower partial pressure of oxygen or a higher partial pressure of oxygen. If we represent this on the graph, achieving hemoglobin saturation with a higher partial pressure of oxygen would mean that the curve shifts right. These conditions make it more difficult for hemoglobin to bind to oxygen, such that even though the surrounding environment is filled with oxygen molecules, it is still very difficult for the hemoglobin molecules to become saturated. You can now see how a higher partial pressure of oxygen is needed for hemoglobin saturation to reach 50%. Or in other words, P50 has now been shifted to the right, indicating a higher partial pressure of oxygen required. Some factors that lead to a right shift includes an increase in temperature, increase in partial pressure of carbon dioxide, and an increase in concentrations of 2,3-diphosphoglycerate. A decrease in oxygen affinity favors the release of oxygen molecules from hemoglobin. So you'll find that most, if not all, of these factors are characteristic of cells and tissues being hard at work. After all, they are the ones who need oxygen the most for ATP production. First, the mitochondria in our cells generate heat when they are busy making ATP. Think of all the energy that your cardiomyocytes, or heart muscle cells, need to continuously pump blood throughout your body. It would make sense for these cells to be higher in temperature and favor a decrease in oxygen affinity, so that hemoglobins can then release oxygen molecules to be used as the final electron acceptor in aerobic cellular respiration. Similarly, an increase in the partial pressure of carbon dioxide is also indicative of tissues being hard at work. Carbon dioxide is a byproduct of cellular respiration and ATP production. So cells producing a lot of carbon dioxide would want oxygen to be unloaded from the hemoglobin molecules. That's why more molecules of carbon dioxide being present also promotes release of oxygen molecules from hemoglobin. Lastly, the presence of 2,3-diphosphoglycerate also causes a right shift. 2,3-diphosphoglycerate, also known as 2,3-DPG, also known as 2,3-bisphosphoglycerate, also known as 2,3-BPG, is an intermediate produced during glycolysis, which, as you may know, is the process of breaking down glucose so that the resulting pyruvate can be used to generate ATP. Again, it is when cells are hard at work at generating energy that the concentration of 2,3-DPG is typically increased in the surrounding environment. These are all factors that decrease oxygen affinity and move the oxygen-hemoglobin dissociation curve to the right. But there are also factors that shift the curve to the left. These conditions increase oxygen affinity, making it easier for hemoglobin to bind to oxygen. In other words, they allow for hemoglobin to more readily bind to oxygen such that fewer molecules of oxygen are needed in the surrounding environment for each hemoglobin to bind to 4 oxygen molecules. Achieving hemoglobin saturation with a lower partial pressure of oxygen would mean that the curve shifts left. Now you can see how a lower partial pressure of oxygen would be needed for hemoglobin saturation to reach 50%. P50 has now shifted to the left. Take pH, for example. An increase in pH, or a decrease in hydrogen ions, increases the oxygen affinity of hemoglobin proteins. This is somewhat related to carbon dioxide as well, because hydrogen ions result from the deprotonation of carbonic acid, which is a product of carbon dioxide reacting with surrounding water molecules. In short, though, the more carbon dioxide that is released from metabolic activity, the more hydrogen ions there are, which increases the acidity and decreases pH. So we can say that an increase in pH causes a left shift, or an increase in oxygen affinity, and an increase in the concentration of hydrogen ions causes a right shift, or a decrease in oxygen affinity. Another factor that causes a left shift is the presence of carbon monoxide. Hemoglobin has a much higher affinity for carbon monoxide, roughly 200 to 300 times more than oxygen. So there are 2 things that happen. 1. Carbon monoxide can act as a competitive inhibitor of oxygen in competing for the same heme groups on hemoglobin. 2. In terms of oxygen affinity, when one carbon monoxide binds to a hemoglobin, it induces a conformational change in structure that increases the oxygen affinity of the other 3 heme groups. The result is a left shift in the curve. The increased oxygen affinity that carbon monoxide binding induces can be so significant that oxygen cannot readily unload from the hemoglobin. This can lead to a severe lack of oxygen in the tissues, which results in the symptoms associated with carbon monoxide poisoning. To summarize, the factors that result in a left shift of the oxygen-hemoglobin dissociation curve include increased pH and presence of carbon monoxide. These increase hemoglobin's oxygen affinity. Factors that result in a right shift include increased temperature, increased partial pressure of carbon dioxide, presence of 2,3-DPG, and an increased concentration of hydrogen ions. These decrease hemoglobin's oxygen affinity. Some helpful and quick ways to help with memorization include remembering the left shift as being associated with oxygen lingering on the hemoglobin, and the right shift as being associated with oxygen being released from hemoglobin. A common mnemonic that is helpful in remembering some of the factors include cadet face right, where carbon dioxide, acidity, 2,3-DPG, exercise, and temperature are all factors that result in a right shift when they are increased or present in the surrounding environment.